Spectrophotometers measure the intensity of light absorbed after a laser passes through a sample solution. This reading provides a quantitative analysis by determining the concentration of a colored solution. Qualitative data can be obtained after the concentration has been found to identify the identity of a solution using the characteristics and concentration known. One would scan across all wavelengths to find the lambda max, which is the maximum absorption occurred. The lambda max is needed to ensure high sensitivity and to minimize deviation from Beer's law. The Beer-Lambert law is A equals epsilon times L times C, representing the relationship between absorbance and concentration. A is the measured absorbance, epsilon is the wavelength-dependent absorptivity coefficient, L is the path length, and C is the concentration. One can manipulate this equation to C equals A divided by epsilon times L, which can determine the unknown concentration by using the wavelength and the absorption obtained from the spectrophotometer. It is important to zero the machine using a blank to measure the absorbance of a solution. A blank contains the solvent without the substance that is being measured, so it can be used to calibrate the machine. This accounts for any absorbance changes made to each substance. After obtaining the proper dilutions for the lab, we transfer the solutions into the cuvettes using transfer pipette. This will be done for each dilution, which we will put into the UV spectrophotometer. Before placing any cuvette into the spectrophotometer, we must wipe the sides to ensure no fingerprints are left on the cuvette. This ensures an accurate result is obtained. Now we put the blank into the machine, placing it with the clean sides in the beam. Here we zero our blank. We take out the blank and close the machine. Next we take our next sample and scan the absorbance. We repeat these steps for the remaining samples. It is essential to record the absorbance of each sample in our notebook. After we have finished the experiment and obtained the proper readings, we dispose of our solutions properly and clean our glassware.